Hi, everybody. Welcome. This is Dr. Izabella Wentz, PharmD, author of Hashimoto's The Root Cause. I'm really excited to have Dr. Richard Horowitz here with us today. Dr. Horowitz is a board-certified internist in private practice in Hyde Park, New York. He's the medical director of the Hudson Valley Healing Arts Center, an integrative medical center which combines both classical and complementary approaches in the treatment of Lyme disease and other tick-borne disorders. He has treated over 12,000 chronic Lyme disease patients in the last 27 years, and his book, Why Can't I Get Better? Solving the Mystery of Lyme and Chronic Disease, was a New York Times bestseller, which is an amazing book. If you guys don't have that, please be sure to pick it up. His book explains his full classical and integrative approach to helping those stricken with tick-borne diseases and resistant chronic illnesses. His website is cangetbetter.com. And you can find him on Facebook. So facebook.com/Dr. Richard Horowitz. And that's Horowitz. So welcome, Dr. Horowitz. So great to have you here.
Yep, glad to be here.
I guess, let's start off with just going over the connection between Lyme and Hashimoto's. As you know, I specialize in Hashimoto's, and my big passion is to help people find their underlying triggers for the reason why they may have an autoimmune disorder. For some people, the root cause is often an infectious one. And I've found that some clients, when you're able to remove the infection or treat the infection, will see a complete remission of their Hashimoto's. Can you talk a little bit about how Lyme disease can trigger autoimmune disease?
Sure. So Lyme is a bacteria. It's a very tricky bacteria. It doesn't just cause inflammation in the body, but it does stimulate autoimmune phenomena. So one of the ways that it does it is there are blebs. These are partial pieces of DNA that are secreted from the organisms, and they're very highly stimulatory to the immune system. So what happens is that some of these blebs go inside the cells, and they cause tremendous overstimulation of the immune system, causing autoimmune markers to appear. So for example, when you look at the scientific literature, you'll see Lyme patients have a lot of positive anti-nuclear antibodies. So they don't necessarily have lupus. If you check their double-stranded DNA, that may be negative, because that's a marker for lupus. And when you treat their Lyme, that ANA may turn negative. So we took down the inflammation. It's the same with rheumatoid arthritis. We see a lot of people with rheumatoid factors that are positive with Lyme, but there's CCP negative, which is one of the clear markers for rheumatoid arthritis. You treat the Lyme, the rheumatoid factors turn negative. And then we have a lot of thyroid antibodies. You get antithyroid antibodies, antithyroglobulin, antiganglioside antibodies, where it affects the nerves. And people complain of neuropathy with tingling, numbness, burning sensations. Anti-myelin antibodies, which you can see in multiple sclerosis. And even in MS, they have found cystic forms of Borrelia in the spinal fluid of people with MS. And you can get antiphospholipid antibodies, anticardiolipin. So the problem with this is that many doctors are thinking that it's a bacterial illness and that it's separate from autoimmunity. But the fact is it's that a bacteria that definitely does cause autoimmune reactions. And I have seen people turn negative with their autoimmune manifestations once the bacteria are properly treated.
Wow.
So I would say, the blebs is one mechanism. Another mechanism is there are these parts of the bacteria on the outside of the bacteria. They're called lipopolysaccharides, or LPS. And we know that these are also very stimulatory to the immune system. So these also will cause a lot of inflammation in the body. And there's something inside the cells, inside the nucleus of the cells. It's like a switch that gets turned on by lipopolysaccharides, and it stimulates this inflammatory response. So not only do the blebs, the partial DNA, cause the response, but the LPS, the lipopolysaccharides, the outside of the bacterial membrane, also stimulates the same inflammatory response. And what we also found is that the people who come to see me, they don't just have Lyme, they also have viruses. And they've now shown that viruses like CMV has now been shown to stimulate problems with rheumatoid arthritis. Or they found that herpes zoster, shingles, has now caused problems with inflammation in the arteries called an arteritis. They recently linked up in the literature this past year Epstein-Barr virus with multiple sclerosis. So what's happening is it's bacteria and it's viruses. And then they've shown this past year that BPA, bisphenol A, has also now been shown to cause autoimmune reactions. as well as asbestos and nanoparticles in the air with pollutants. They're causing it, and we see a lot of mercury showing up in our patients, which causes hundreds of autoimmune reactions in the literature. And finally, the microbiome of the gut, the bacteria in the gut may be thrown off, and they've now shown that there are these specific bacteria that look like they're also causing autoimmune reactions. One of them is called Prevotella and Clostridium, and they've shown this with rheumatoid arthritis. And there's one called Firmicutes that's been shown to also cause inflammation and affect methylation in the body and how we get rid of toxins. And with MS, they recently published Clostridium species. Clostridium perfringens has been shown to be associated with autoimmune. So my take, and the model I use for chronic disease is that it's never one thing. I in my book, it's referred to as the MSIDS map, that there's up to 16 reasons why people get sick. So the bacteria, the viruses, the toxins, the gut with the microbiome, these have all been linked up to autoimmunity, but Borrelia specifically has been definitely one of those triggering factors that makes all of the other factors worse at the same point in time.
Wow. So it's kind of like a perfect storm that basically confuses the immune system. And you've got a lot of things happening with molecular mimicry when there are different proteins on the bacteria or viruses or pathogens that may look similar to the proteins that we might find within our own body, like the thyroid peroxidase or the thyroglobulin proteins. And
right. I mean, the molecular mimicry, it's an important point. I'm glad you brought it up, because what happens is the bacteria have tails called flagellum. And your immune system tries going after the bacteria with these tails. And instead, sometimes it looks like the myelin sheath in your body. So what happens is, in trying to kill the bacteria, it ends up causing demyelination. And that's what we see with these people who have very bad neuropathy, who have multiple sclerosis type symptoms, where it's not true MS, it's Lyme mimicking an MS type presentation. So between the molecular mimicry and the blebs and the LPS and the virus, that's all the things, that's the perfect storm, as you're saying, that's causing a lot of these autoimmune reactions in people.
Yeah, and I like to think of basically the immune system taking little snapshots of parts of the bacteria and then showing it around, but then maybe the picture is not as clear. So then there's a little bit of confusion, and then, basically the immune system acts something that looks a little bit like that picture, but is not quite the same. So you've called Lyme a great imitator.
Yes.
Yeah. And
so you'd like me to explain that a little bit, maybe?
A little bit, yeah, just a little.
So the problem is that we have all these chronic diseases like chronic fatigue syndrome, fibromyalgia, autoimmune diseases, MS, lupus, rheumatoid. What happens is that you get a lot of inflammation from Lyme. And what they've shown in a lot of these different diseases is the underlying common denominator is inflammation. There are these inflammatory molecules called cytokines, interleukin-1, interleukin-6, TNF-alpha, interferon gamma. So all of these inflammatory molecules are seen with fibromyalgia, with chronic fatigue, with autoimmune. And Lyme causes the production of all of these inflammatory molecules to take place. So what happens is chronic fatigue and fibro, there's no diagnostic test at this point that's a good test to prove it. It's basically people are tired, have joint and muscle pain, they can't fall asleep, their memory's off, and they may have some autonomic nervous system problems with low blood pressure. But those are the same symptoms you see with Lyme. So it's the great imitator, because if people just do a test like an ELISA test, which is what they've sometimes been taught to look for Lyme, and the ELISA is negative, they say to them, oh, well, you have chronic fatigue, you have fibro. Or you have just simply an autoimmune disease, you don't have Lyme. But because the testing is so inaccurate, there are over 100 strains of Borrelia in the United States and over 300 strains in the world. There's no way that this one blood test, the ELISA, is going to pick up all of these strains. So it's a great imitator because it's imitating all of these different diseases, but really the underlying common denominator is inflammation causing all of the same symptoms that we're seeing in these different diseases.
Okay. Well, thank you so much for explaining that. And I'd also love to find out from you, how does Lyme specifically affect the HPA axis?
Right. So when you get these inflammatory molecules produced, what happens is these inflammatory molecules shut off the hypothalamus and the pituitary axis in the body. So what we find happening with the hormones is I might, for example, have young men in their 20s coming in with no testosterone, like they're 80 years old. They've got a testosterone of 150 or 200 because the luteinizing hormone has been shut off from the pituitary. Or women, the FSH, the follicular stimulating hormone, may be shut off and they're not getting their periods. we see about at least 40% of our patients, the adrenals are very, very low. The ACTH, the adrenal stimulating hormone, may be shut off. And then with the thyroid also, we can see different problems with the thyroid where they will get hypothyroidism from the antibodies, or they simply may have a lot of cortisol produced from too much stress and that converting T4 to T3, or they're getting too much reverse T3 because they're missing selenium or certain minerals. So all in all, the hormones basically are affected by the Lyme. So it's not enough to just treat the infections. You've also got to affect and kind of balance out the damage to the body that's happened from the infections. So part of that is fixing the hormones. And a lot of people don't realize that the hormones are affected in almost half of the people, at least that I see with Lyme. And it does make a big difference when you get the hormones back in balance.
Wow. And I know I see people on the other side of things where they will actually think they have a thyroid disorder, but really the reason for their thyroid disorder is because they have Lyme disease. Can you talk about how a person would know that they have Lyme disease, what kind of symptoms to look for, as well as the best testing that you recommend? Because I know a lot of tests out there can produce false negatives, right?
Right. So the problem is that when they started looking at the tests years ago, they were using this one test called an ELISA. And the ELISA is supposed to be, a reasonably good test to pick up Lyme. But as I said earlier, it will not pick up all the different strains of Lyme. And we're now even getting European strains like Borrelia abszelii, Borrelia guarini. There are relapsing fever Borrelia like Borrelia miyamotoi. It's a relapsing fever from Japan. It's not going to be picked up by the ELISA. So there's a newer ELISA called the C6 ELISA. It's a better test. It has about an 88% sensitivity, although when I've checked it, I don't think it's quite as high as the literature, but it's definitely better than the ELISA. That is certain. But I find that when you look at the literature on the ELISA, it only picks up about half the patients who have Lyme. So what I do is I do a Western blot. But the problem with the Western blot is that the Western blot will only pick up, again, a certain percentage of all the different strains of Borrelia. So, for example, Quest or LabCorp or BioReference, some of the big reference labs, they use one strain called the B31 strain. Well, again, if you have over 100 strains in the US and 300 strains in the world, you're again not going to expect that a Western blot from a localized lab is going to pick it up. So the way that I diagnose the disease is not to use the two-tiered testing of an ELISA followed by a Western blot. The CDC themselves have said that's really only for health departments to epidemiologically screen large populations of people. Even the CDC has said it's a clinical diagnosis. So the question then is, well, how do you make the clinical diagnosis? Well, there are 6 signs and symptoms that I find are usually the most specific ones for Lyme. So they are, for example, that number 1, the symptoms tend to come and go. You have good and bad days. Okay. The joint pain tends to migrate in the body. The muscle pain will migrate. Even the nerve pain, the tingling, numbness, burning, or stabbing sensations, that will also tend to come and go and migrate. And migratory joint pain, muscle pain, or nerve pain is very specific for Lyme. So if people tell you they have good and bad days and it's migratory, that's very specific. Women will tell you around their menstrual cycle, just as the estrogen is coming down, that the Lyme starts coming out. So that's also a clue in women. Sometimes around the hormonal cycles, you can see it coming out. If people take antibiotics and they're unaware that they have Lyme, a lot of times their symptoms of what they thought was chronic fatigue or fibromyalgia gets better because you've knocked down the bugs, but sometimes it gets worse. That could be from a Herxheimer reaction, which is when you're killing off the bacteria, you get release of these inflammatory molecules, right? And then when you look at the Western blots, and you look at the bands on the Western blot, I like calling this Lyme bingo. There are 5 bands on the Western blot, and any one of these bands that I'm about to tell you are very specific for Borrelia burgdorferi, but also will show up in other strains of Borrelia. So these are the following numbers: the 23, which is the outer surface protein C, the 31, which is the outer surface protein A, the 34, which is the outer surface protein B, the 39, and the 8393. If you have any one of those numbers, 23, 31, 34, 39, 83, 93, with the symptoms of coming and going, good and bad days, migratory muscle, migratory joint, migratory nerve pain, changes around the hormonal cycle, better or worse with antibiotics, that basically tells you that you have a clinical diagnosis of Lyme. And we found that to be quite sensitive, and then I've developed a questionnaire. And this questionnaire is in my book and it's on my website. People can just go on cangetbetter.com and take it. And this past year, we had SUNY New Paltz, the State University of New York, looked at the questionnaire to validate it to see if it was valid statistically. And we actually found that it validated out in about 500 people, both with Lyme and without Lyme. If you have a score of over 46 on the questionnaire, it means you have a high probability of Lyme. So if you put together the questionnaire of having a high score, having any one of these Borrelia-specific bands, and looking at the symptoms of coming and going, you've got a good idea whether you may be looking at Borrelia, especially if you've ruled out other diseases. That's basically how you can help make the diagnosis.
Okay. Thank you for that, Dr. Horowitz. And I've been sending people to your questionnaire for probably a little over a year and a half now. And so a score above 46 for people to remember, and I'll put up the link for that. Thank you for going over those. One of my friends, colleagues, one of the things he talks about, Brent from Lyme Less Live More, is that some people have some odd kind of, symptoms, like one of them being sensitivity to Wi-Fi. Have you heard of that? And could you offer any kind of guidance on why that might be the case?
Yeah, so I see a lot of chemically sensitive people who come to see me. Basically, we're exposed to over 300 environmental chemicals every day. It's everything from pesticides to xenoestrogens that are causing all forms of cancer. A lot of people, their detox pathways do not work well when they have Lyme disease. Mm-hmm. So when you get all of these chemicals in your body and your detox pathways aren't working, and then on top of that, you start to get some people with Wi-Fi sensitivity who are sensitive to EMFs. The EMFs also disturb the cell membranes and cause inflammation in certain people who are genetically, they just are wired, kind of, no pun intended, to be that way. I wouldn't say that I see a lot of it in people, but I definitely do some of it, see some of it where people are that sensitive. And I find that if you can lower down the inflammation, in the MSIDS map, there are multiple factors where you can lower the inflammation in the body. So I find that, for example, if you work on diet, let's say you're sensitive to certain foods, and you're eating gluten and you shouldn't be eating it, or you have certain food sensitivities, that will help lower inflammation. Or if you're not getting to sleep, that causes inflammation with interleukin-6. Or your zinc is off, your minerals are off, that will cause inflammation. Or you have co-infections. I find the co-infections, Babesia, the parasites. So what happens is it is a perfect storm. You've got all these infections, all of these overlapping factors, like your gut is off, you have dysbiosis in your gut, you're not sleeping, you have heavy metals and all of these toxins getting in. Those are the people I find that have the most difficulty with the chemical sensitivities. And I find that, some people have actually had to use lead shielding to be able to get better. Fortunately, I haven't found it to be that bad in the majority of my patients, but certainly some have been bad. And it does make a difference once you can unload their body of the toxins and once you can lower all of these different ways that they have inflammation in the body. So you've got to, again, look at why these people have an inflammatory response. Because if you look at all chronic diseases, all of it at this point has linked up, whether it's Parkinson's, Alzheimer's, heart attack, strokes, asthma, scleroderma, every medical condition that you look at, when you get to the underlying causes, it's been free radical, oxidative stress, inflammatory problems. And I don't think anybody has ever looked at it from a multifactorial cause. So if they have Wi-Fi sensitivity, usually they're going to be chemically sensitive. And these are the people that usually need to go do saunas and looking at the pathways in their liver to start unloading the toxins and cleaning up their diet, cleaning up the environment around them. Those are usually the people that will get the most benefit from looking at all the sources of inflammation in the body.
Okay, thank you. That was very helpful. And I like to think of, getting your ducks in a row and trying to do all these things, and kind of like if you were to adjust switches to get them right in the middle. So you're optimized on your nutrients, you're eating the right kind of diet for you, and so on and so forth. But, one thing that I think a lot of people have questions about, and, the whole chronic Lyme thing is a controversy. And of course we know that Lyme is caused by a bacteria, and Borrelia, and it's a type of bacteria, and we have antibiotics, right? So why is it so hard to treat and get rid of Lyme disease?
Yeah, it's a great question. So the problem with it is that it can be cured if you get it early. Mm-hmm. So, but the problem with early Lyme, in fact, I can go back to the testing for a second. The problem right now is the antibodies are not produced for at least several weeks, even on the IgM antibodies. So it could take at least 3 or 4 weeks before you start getting IgM. So there's a test now that they're working on, and it's come out of Johns Hopkins researchers. His name is Solosky. And they found that there are these inflammatory molecules in the body. They've got fancy names, CXCL9, CXCL10, CXCL19. They're now doing a test at Columbia University called the Spira test, where they're looking at these molecules. So Lyme is actually easy to treat if you can catch it early. But the problem is the testing we're using doesn't catch it. Hopefully, some of these newer tests will be able to pick it up. So that's the first thing. So if you have a tick bite and you're not sure whether it was on for a while, because the problem is when you look at the literature, the European literature, a lot of people think it's got to be on for 24 to 36 hours. The European literature, there's literature of 16 hours, some even 6 hours. The Powassan virus, there are viruses in the ticks that can get in 15 minutes. Yeah. Certain rickettsial infections like Rocky Mountain spotted fever can get in within 10 minutes. So people need to know you can get tick-borne infections without the tick being on for very long. But if you're not sure, it is sometimes better to prophylactically to be treating with doxy or tetracyclines early on in the illness. And even kids, they've now shown short courses are okay even in children. So if you treat it early, it's curable. But what happens in chronic Lyme is that the Lyme will start to change forms. So if you think of the Lyme as having an outer surface protein, it's got these cell wall forms. It also can go inside the cells, inside the intracellular compartment. Now, we know that these intracellular bacteria, they're very difficult to kill, but it also can form cystic forms. They're sometimes called cell wall deficient forms, S forms, L forms. These cell wall deficient forms is one of the ways that it persists in the body for very long periods of time. We also now know that Lyme will cause biofilms. And some of our most chronic, most difficult to treat infections we now know are in biofilms. So the NIH has found that 60% of most chronic infections and 80% of difficult to treat infections can be due to biofilms. So the problem we face is we only learned about biofilms in Lyme just in the last couple of years. And there are different biofilm busters like nattokinase, Baluch, EDTA, serrapeptase, and there are newer biofilm busters like stevia, and they found that like pomegranate extract and maple syrup, there are like these new ones that have come out, but the problem is nobody's tested them to know the best combinations. So when you think about it, if you don't treat all the different forms of the Lyme, if it's constantly changing form, and let's say you only gave doxycycline, doxy mainly goes inside the cells. But other drugs like Rocephin or cell wall drugs like amoxicillin or Ceftin, those are outside the cell. They don't penetrate inside the cell. So some of the ways that I've had great results with treating is to realize that the biology of the organism is it keeps changing forms. So you've got to treat the cell wall forms. You've got to treat the intracellular forms. You've got to treat these chronic persistent cystic forms. You've got to go after the biofilms. Right, so you've got to go after all these different forms to be able to get it. And also the co-infections are very important. I have found that over 90% of my patients have co-infections like Babesia, which is a parasite. So if anyone is listening and they end up having day sweats, night sweats, chills, flushing that they can't explain, and they're not, perimenopausal or menopausal, or they have a cough they can't explain with air hunger where they can't catch their breath, those can be symptoms of Babesia, which is a parasite. If you don't treat that parasitic infection, it's much more difficult to treat the Lyme. So when I'm looking at getting people better from Lyme and why it's so difficult to treat, I have to kind of go back to this 16-point MSIDS map, multiple systemic infectious disease syndrome, which is it's bacterial infections, it's viral infections, it's parasitic infections, it can be yeast and candida, it's autoimmunity, it's inflammation in the body, it's being on the wrong diet, it's not getting enough sleep, it's the hormones being thrown off, right? It's all the toxins getting into our body. If you don't address all of these different 16 points, I find that people don't get better. So you can't just treat the infections. You've got to look for all of these overlapping causes of why people may stay sick and have inflammation in the body. But I do think that the biofilms is going to probably play a very important role of why we've had difficulty. And John Hopkins and researchers at Northeastern University, Kim Lewis recently, they've been doing studies on pulsing antibiotics and have found that at least in culture, if you pulse the antibiotics, you might be able to kill Borrelia. The problem is in the studies they've done, they're kind of young cultures with the Borrelia actively replicate. In people who've had Lyme for a while, that may not be the case. And there are also persister drugs. Dr. Zhang from Johns Hopkins, I've been working with him now, and we're looking at persister drugs, newer drugs that people haven't used, like daptomycin, clofazimine, sulfa drugs. And I found that some of the new persister drugs that I'm using in my practice, I'm actually having very good success. So I will hopefully be getting some research out very soon on these, but I've started to use these regimen, and even in some of the people that have not gotten better with my prior protocols, I'm finding that they are getting better. And there may be a new book, if I can get this out by next year. My publisher is having me work on this now. Instead of why can't they get better, we're looking at calling it how can I get better? And it's going to talk about all the new science that's come out in the last couple of years with pulsing and persisters. It will be all the updated science on why people don't get better from Lyme and what the science has taught us. But there's some very exciting research. The university researchers are finally getting involved and finally doing something looking into causes. And that has not happened actually for years.
Well, I'm really excited to hear that the university researchers are now getting involved in your work. So very grateful for that. So basically Lyme, the Borrelia bacteria can actually mutate and it changes forms so that the traditional antibiotics are not going to be effective against it. So we have to attack it from a variety of different places. And it also kind of creates a community for people not familiar with biofilms. That's basically like a community of different pathogens and kind of, slime, for lack of a better word, that different kind of pathogens build around themselves to protect themselves from maybe antibiotics are not able to fully penetrate that. With your experience, you have, some patients that actually do get better and you can eradicate the Lyme and reverse the associated immunity.
Yes. Well, the thing is we don't know at this point. I mean, the patients, I get the vast majority of my patients better, but the problem is that some get a little better, some get moderately better, some are fantastic, and some have no symptoms at all. But my concern with the Borrelia is that it still may be persisting in very small quantities. But I don't know that we need to eradicate every last bacteria from the body. I mean, we have, as you know, we are made up of 90% bacteria and we're 10% human. So when you look at the microbiome of the gut, we're mainly made up of bacteria. We're not really mostly human cells. So I don't think it's possible or even necessary to get rid of every last bacteria. What I think is necessary is to form that kind of symbiotic relationship of how we live with the bacteria. I think it's necessary to lower down the load of the bacteria in the body. And I think the people that I've had the greatest success with, those are the ones where we balance the autoimmune reactions and we've detoxed them. I mean, the detox is absolutely essential because when all of these inflammatory molecules, these cytokines are being produced, if you don't get them out of the body and detox them, they don't feel better no matter how many herbs or antibiotics you throw at them. So I think really it's a question of kind of getting the bacteria in balance in a symbiotic relationship. And I think the future is really going to be the microbiome is figuring out. There's a lot of new research on Parkinson's and Alzheimer's with the microbiome and autoimmunity on it. And some very exciting things. Same thing with stem cells of people who have been damaged where stem cell therapy may be able to reverse some of it. So I think that's going to be some of the future of how we're looking to get people better. But fortunately, over 90% of people get better with the 16-point MSIDS map. But what's exciting for me is the people who didn't get better, like the ones who've been coming to me for years who've said, Doc, I get better on your protocols, whether it's herb antibiotics, but I relapse off and nothing gets me better. Some of these newer persister drug regimens that I've been doing in the last year, I have people coming in who failed every regimen who are getting better. And I hope some of these relate to like mycobacterium, almost tuberculosis type regimens that no one has been using. And I've had some amazing successes. We just don't know, will the success persist? How do we get the drugs, the right dosage? So I'm working on it right now. I have a lot of patients entering into these treatment protocols, and I'm working with Johns Hopkins researchers right now. But I think that if you look at the 16-point map and you treat the parasites and all the bacteria, they get better. But I really think the future is going to be, we've got to do better research on the biofilms where we can't get to the bacteria because of the biofilms and where they're hiding inside the cells intracellularly, because a lot of these new drugs that I'm using are going inside the cells to kill the bacteria. And some of these resistant patients are really having miraculous results. So I've been speaking to Dr. Zhang at Hopkins and letting him know about this. And he's going to take some of these protocols now and go into the lab, put them in culture. So I finally have a working relationship with university researchers so we can actually look at this. And it's really an exciting time.
Yeah, I'm super excited to hear that. And as a pharmacologist, that just kind of gets me excited to hear about all the new protocols that are going to be available. Can you share some of the protocols that you utilize within your own practice that you found to be the most helpful? Even just maybe some detox protocols, and you talked about nattokinase and any kind of antibiotic protocols that people might, will need to work with their physicians on, of course, as well as
Right.
what they could do on their own.
Sure. So, the thing is that a lot of the herbal and natural protocols, they do work. I find that probably on the average, about 70% of people who take the herbal protocols do notice that they get help. So, for example, Stephen Booner has done some very good work, looking at andrographis and Japanese knotweed and smilax and a lot of these different herbs, cemento. It definitely works. The problem is some of the people who are super sensitive, they have like HLA-DR2 or 4. They're genetically wired to have very strong reactions. They sometimes have problems with cat's claw on some of these herbs. But we've used, for example, Chinese herbs, traditional Chinese herbs, like from Dr. Zhang, Coptis, Hoodiana, R5081, Artemisia for Babesia, Neem for Babesia, Cryptolepis for Babesia. So many of those protocols have been very helpful for these parasitic type of infections. In some people, even homeopathy has worked for some. Some of the classical homeopaths have had some luck. We use a lot of the Byron White protocol, using AL for Lyme, A. Bart for Bartonella, A. Myco. Some of his protocols are very strong. People can be, 1 or 2 drops of this stuff is enough for some people. They're that sensitive to it. The Beyond Balance protocols from Susan McCamish, BB1, BB2. We basically rotated through a lot of these herbal protocols, and we found that, in people even that can't take a lot of antibiotics because they may have candida, gut issues, they herx, we can sometimes even put them on 1 antibiotics and then just give them herbs. So we use, a combination of classical and integrative, and that works actually quite well too. But the detox, as you said, it's really crucial. So for example, glutathione, there are 6 pathways in your liver to get rid of toxins, and one of them is glutathione. And glutathione takes fat-soluble toxins in your body and makes them water-soluble so you can basically urinate them out of your body. And I started doing studies on glutathione years ago when people were having brain fog and memory problems. And about 70 to 75% of my patients say that they will get good help with glutathione. And there was, in fact, one person who came in about a week ago. He was sick in bed for about 2 years. He's a computer specialist. He used to actually work on programming. He couldn't work at all from the Lyme. Every regimen he did herxed him or didn't work. The first time in my office, I gave him a 2,000 milligram, 2-gram shot of glutathione. His brain completely cleared because we were probably pulling out neurotoxins. So again, the problem with the Lyme is it's not just the bacteria causing inflammation. There's also neurotoxins that are getting in from the Lyme. And one of them is called quinolinic acid. So when you look at all the toxins getting in from the environment, we're finding mold toxins in about 75% now of our patients. Up to it are now finding ochratoxins, aflatoxins, trichothecenes. Real-time labs in Texas, we've been finding, about roughly 70% of these people who say, Doc, I'm not responding well. They have high levels of mold toxins. So you've really got to detox these people using phosphatidylcholine, with glutathione, with alpha-lipoic acid, and butyrate to push these toxins out of the body. Sometimes people have methylation defects. They're MTHFR positive. So we have to give them more methylation support. So we find that by working the pathways through methylation, through glutathione pathways, lots of water, right? Dilution is the solution to pollution, right? I didn't make that up. That's Sherry Rogers. Doing infrared saunas to unload the body of toxins, especially in the chemically sensitive people. A lot of these detox protocols are very, very helpful. So if people were to use some of these natural protocols and detox, we do find that actually it makes a big difference, and of course, cleaning up their diet, getting off their allergic foods, getting to sleep, getting onto good exercise programs, even physical therapy programs. People can do that on their own without having, of course, to take pharmaceutical drugs. So there's a lot that people can do, and hopefully by the end of the next year, we're working on an app called the Lyme Navigator app. And the foundation is a 501c3 called the Lyme Navigator Foundation. And I'm working with a computer specialist, David Turek, and we're working on this. He used to work for Bell South Labs, and David and I and his brother, Drew, were working on this app so that instead of coming to see me, you can go on your phone or go on your computer, put in your symptoms, put in all your medical history, all your lab tests, and it will give you a differential diagnosis and then tell you what tests you might want to do with your doctor. You then put those tests in, it will give you options, and you can work with either your naturopathic doctor or your regular MD, and it will give you protocols. And we're going to do this so that thousands, I mean, literally millions of people can do this, where it can be uploaded into the cloud. So if we have protocols, let's say that we want to try in multi-center studies, within months we'll be able to see how effective these protocols are and start to move through it, because Lyme is the #1 worldwide spreading epidemic, right? So it's the #1 infectious disease. When I was in China years ago and the Chinese government called me over and invited me over, they told me that 6% of the Chinese population had Lyme.
Wow.
So we're finding that huge numbers of people worldwide are being infected with these bacteria. And we need to find ways to quickly and efficiently go through protocols, because we are having problems at this point with it. So hopefully the Lyme Navigator app will solve some of this problem. So hopefully by the end of the next year, we'll have this app going, and hopefully I'll be able to share more with you at that point. But again, this will hopefully solve the problem, because I can only treat so many people myself. That's why the book was written. I was hoping to share all my secrets, with everyone. And then I found out that it still was too complicated. So David and I said, all right, let's do an app. I'll do a simpler book. How can I get better? It'll be a soft cover, much easier, not exactly MSIDS for dummies, but much easier, not encyclopedia of medicine book kind of to get this. So I realized that we have to keep working on this, but it will have all the updated science. And as we keep discovering new things, we can upload this in the app. So like the most up-to-date treatment protocols will be immediately available to people. So I hope that this in the next year or two will kind of have this in place.
Wow, that sounds really amazing. So to give people protocols at their fingertips that they can use on their own, as well as take to their physicians to work on some of these antibiotic protocols as well. So so you mentioned the MAP infection, Mycoplasma avium paratuberculosis. Do you see that as a co-infection in people with Lyme disease, or?
What's interesting is that I think there are a lot of from what I can tell, the intracellular bacteria are some of the ones that are actually causing the biggest problem with this disease. So, for example, people test positive for Mycoplasma species. Years ago, when we did a retrospective study, we found that about 13% of our patients tested positive for Mycoplasma fermentans, which is an intracellular bacteria. They have chlamydia pneumonia, which also can have other manifestation of causing inflammation. As far as the mycobacteria go, it's kind of tough to know how many people really have mycobacterium. But the reason I took an interest in looking at the protocols is that a lot of these bacteria are slow-growing, intracellular, difficult to kill, the exact same type of bacteria that are mycobacterium. And when Dr. Zhang at Hopkins was looking at persister bugs and persister drugs, some of the mycobacterium drugs like pyrazinamide with rifampin. Now, we've used rifampin for Lyme and Bartonella for years. So my interest was to see whether some of these mycobacterium regimens would actually be useful. And I'm finding that, it's possible that some of these people actually have mycobacterium and we don't even know it. So one of the things that I'm discussing right now with the Hopkins researchers with Dr. Zhang is actually getting blood over to him to see whether we can actually test people, because in the laboratories that I'm using, I don't really have a way to test, and I suspect that people may have many species, for example, of mycoplasma. They may have mycobacterium. They may, Bartonella, there's over 30 species of Bartonella. Regular labs, they check Bartonella henselae, maybe Quintana, Bacilliformis. I can't use, for example, Galaxy Labs in New York, which checks for over 30 species. So I need good researchers at universities to tell me in my patients that are not getting better, how many strains of pyroplasms of Babesia of parasites do you have? How many Bartonella species do you have? How many Mycoplasma species do you have? Do you have Mycobacterium? I suspect that these are multiple infections. I already know they are in some of these, but I can't really validate the full number because we just don't have, the laboratories that can tell us. But hopefully, as we develop better communication with these university researchers and doctors, in the trenches like myself, it's really the first time that this has started to happen because the politics finally, after all these years, are starting to change. I mean, thank God, after all of these years, people have realized it's like time to put down the guns. Let's stop shooting at each other. Even the Infectious Disease Society of America, from what I've heard privately, they're maybe looking to get a ILADS doctor, on their committee to look at the guidelines, because their guidelines are 9 years old. They've not been updated. There's a lot of new science that's come out, and it's really old science. So, hopefully we're going to start to work together. And when I've discussed it with some of these people from the other side, including Ben Beard from the CDC, I met with one of the heads of the CDC last year. And I said to him, the way around this is all the prior research was done on Lyme. Nobody was looking at Lyme and co-infections.
Mm-hmm.
And I'm finding that these co-infections are just playing a huge role, including the parasites like Babesia. And I do suspect we are probably going to find Bartonella and Mycoplasma and maybe Mycobacteria will show up. But until we put aside the politics and we have those university researchers working well with us and we get the funding that we need to do the studies, I mean, this is a worldwide epidemic. We've got to put our resources and all of our time and effort into this because it is imitating all these different diseases, and it's in the blood supply. Babesia is in the blood supply, Anaplasma is in the blood supply, Bartonella is in the blood supply, relapsing fever bacteria. So when you get a blood transfusion, they've now discovered that 4 out of 1,000 transfusions have Babesia in the United States.
Wow.
And the Red Cross is not screening. And women can transmit some of these infections to their children. So you're looking at the future generations of America where the women are not being screened by their OBGYNs, right? They could be using the MSIDS questionnaire in their waiting room to see if they have a multisystemic disorder. It's not being done. They're not really being given proper prevention tips. So we're talking about all these toxins getting in. We now know with autism. In the last year through Harvard and UC California, Davis, the pesticides and methylene chloride and heavy metals have now been linked up with autism, right? So when you're looking at the autism epidemic and you're looking at the Lyme epidemic and chronic disease epidemics, we're looking at multifactorial causes. It's kind of time at this point to really put our research into looking at this and getting the people in the trenches with the university researchers, right, to be doing this. And it looks like it's finally happening, but my God, it's taken us long enough to get to this place, but I've lived long enough. I'm almost doing this for 30 years. It's finally happening. But I do think that in the next few years, we're going to have big breakthroughs. I suspect in the next 5 years, you will see where some of these persistent drug regimens and pulsing regimens, because I know Kim Lewis from Northeastern is working on this and he's getting funding. I think we're going to see some big breakthroughs in the next couple of years on this.
That's really exciting news. So, Dr. Horowitz, thank you so much for the work that you're doing and taking time out of your busy schedule to come and chat with me to educate people with Hashimoto's about Lyme disease. Where can people learn more about you and your work, and how can they get a hold of you and get in touch with you?
So on my website, cangetbetter.com, there's a place where you can see the schedule of where I'll be speaking. My next big speaking engagement will be at Kripalu. I mean, I'm going to be in Europe, but I don't think people want to follow me to Belgium and France. But I have, maybe they will, but I'm speaking in Antwerp actually in about 2 weeks. I'm speaking to the Belgian government in the middle of that week, and then I'm in right outside of Brussels. But I'll be speaking at Kripalu in Lenox, Massachusetts in December 4th, the weekend of December 4th. There'll be a 3-day workshop that I'm doing with my wife on Lyme disease. So it's a full download of all of the updated science, the 16-point MSIDS map. So on the website, Can Get Better, every time I have these speaking engagements, I was just at Omega. It'll be listed on the website if people want to follow. But between Facebook, I'm posting a lot of the new science on Facebook for the posts. They can certainly follow me, there to see what the new science is at this point. And the Can Get Better website, again, as the new book comes out, I'll let people know about it, but hopefully by the end of next year, that will be out too.
Great. Well, thank you so much for this. I'm really excited about the work that you're doing, and we'll be excited to follow up with you to see, when your new book comes out.
Thank you.